Hey everyone, let's talk about the 3-point cross. So what does the 3-point cross do? What can it do? Well, given three linked genes, it can tell us what gene is in the middle. So, if we know the gene in the middle, it doesn't tell us what gene is on the left or the right of that middle gene. It can tell us what gene is in the middle. And I'm going to walk you through an example 3-point cross. And again, this is outlined in the notes if you'd rather read, but I can also walk you through it. And the 3-point cross is going to pull together concepts that we've been talking about since the first lecture, so I think it's a good way to end this part of the course on classical genetics, at least with respect to eukaryotes. We will have one more lecture that deals a little bit with classical genetics, at least with respect to bacteria, which we haven't talked much about yet. So let me walk you through an example. So in this example, we are going to talk about 3 imaginary genes: foo, bar, and baz. Now, let's say we've discovered these genes. We have mutations for each of these such that the wild type version of each of these alleles is dominant to the recessive version. And we know they're linked on a chromosome in an organism. We could pretend it's Drosophila. The organism doesn't matter. It'd be nice if it makes lots of offspring. So let's say we wanted to, now we know they're linked on a chromosome, but we don't know the order of these genes on a chromosome. So in order to do that, we need to do a cross. And let's say, in the cross, in order to do the 3-point cross, one of the parents must be heterozygous for each gene in the analysis. So I'm going to start like this. You know what? I'm going to go and use our shorthand notation too. I'm going to put plus, 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 and then foo, bar, baz, like that. Okay, so this parent 1, is heterozygous for each of the genes. And parent 2 is going to be homozygous recessive. Almost always going to be homozygous recessive. So, and you should go back, and the point here is that we want the phenotypes of the offspring to match the genotypes of the gametes from the heterozygous parent. And that was a major concept covered, I think, almost in all of Lecture 7. So go back and review Lecture 7 to see why, in this case, we want parent 2 to be homozygous recessive. And just briefly one more time, it's because we want the phenotypes of the offspring from this cross to match the genotypes of the gametes from parent 1. Again, that is Lecture 7 where we cover that. So we want to perform this cross, and then we want to see we want to get the phenotypes of all of the offspring from this cross. Okay, and I'm going to pull up a table that will allow us to see the various phenotypes from this cross. And so one thing, before we look at the phenotypes, we need to predict a gene order. So step 1, we're going to do the cross. Sometimes I call that step 2, but step 1, predict gene order. Okay, now let me address one thing before we go on. We have two different concepts here. We have gene order and we have allele arrangement. So gene order, when I say gene order, I mean if you take a map, a genetic map, you make a genetic map, what is the order of the genes on the chromosome? With a 3-point cross, we don't really know what comes first, what comes last, but we know what's in the middle. So is bar in the middle? Is foo in the middle? In that case, I can put bar here and baz here. Or is baz in the middle? So again, with respect to a 3-point cross, this foo bar baz with bar in the middle, I'm going to put bar here. I'm going to put baz here. I'm going to put foo here. These are equivalent. Bar is in the middle, here and here. That means it doesn't matter that baz is listed first here and foo is listed first here. Those are equivalent. So when I say gene order, I'm talking about which gene is in the middle on the chromosome. Allele arrangement refers to this. So let's assume bar is in the middle. This is one arrangement of alleles. We have all the wild type alleles on one chromosome, all the mutant alleles on the other. Another possible arrangement is that. So bar is with the two wild type versions of foo and baz, and vice versa on the other chromosome. Foo is with baz, the mutant baz, and the wild type bar. We could come up with another one. Foo and bar on one chromosome with the wild type baz, and plus and mutant baz on another chromosome. So these are allele arrangements. Keep in mind, okay, this arrangement here is identical to one that has the pluses on the bottom and foo bar baz on the top, right? Doesn't matter whether we put this on top or bottom, those are equivalent. Same thing if we were to flip this around, baz bar foo, these are equivalent too, right? Because it's only the middle gene that we're interested in. You know, we can't tell the difference with a 3-point cross between this scenario here where baz is listed to the left of bar and foo is listed to the left of bar. So those are equivalent. So this is allele arrangement. Allele arrangement is talking about the arrangement of alleles on the individual homologous chromosomes, and the gene order is referring to which gene is in the middle with respect to the other two genes. So when we're setting up this cross, first thing, the 3-point cross, to figure out which gene is in the middle, we need to predict the gene order. Predict gene order, which one is in the middle. And I am going to predict bar. Bar is in the middle. So, okay, so we do our cross. Step 2 is perform the cross between the heterozygous parent and the homozygous recessive parent in this case, which we just talked about a second ago. So foo, bar, baz, cross to foo, bar, baz, foo, bar, baz. Okay, now what we need to do is collect the progeny and count the different phenotypes. You know, there are going to be 8 different phenotype combinations, and we need to just analyze the progeny and count up the numbers of each phenotype. And the 8 different types are going to come from the 8 types of gametes that parent 1 can make as a result of no crossover, single crossovers, and double crossovers. So I have a table somewhere. Let me get that here. Here's the table. And I will tear this. Okay, so imagine you did this, maybe this is Drosophila, you mated these two parents and then you collected all the offspring. And you know, I have never really worked with Drosophila. I did once in a class 20 years ago or something like that. And I forget how many progeny they made. Maybe we have to cross multiple parents with this genotype with multiple parents with that genotype. But in any case, these are the progeny numbers we get. So I hope this is in focus. Okay. I better tear some more. Oh, I tore it too much. Okay, here we go. It's lying mostly flat. I was like, so we collect all the offspring and we found 370 with this phenotype, which is wild type for foo, wild type for bar, wild type for baz. And 350 for this phenotype, which is mutant for all three. Now, step 3, this is step 2, making this nice table with all the different numbers of progeny in each group. We need to confirm the allele arrangement. Okay, so I made a prediction up here that in parent 1, the heterozygous parent, that all the wild type alleles were linked on one chromosome and all the mutant alleles were linked on the other chromosome. Now, remember, we had determined in the previous video, actually, that non-crossover gametes should be formed in the greatest number. So if this is true, and this is the arrangement of alleles in parent 1, then plus 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 should be one non-crossover gamete, and foo, baz, bar should be found in the other non-crossover gamete. And we also determined that non-crossover gametes should always be found in the highest numbers. So if this is really the allele arrangement, then these are the non-crossover gametes, then progeny with these phenotypes, wild type for all three or mutant for all three, should be found in the highest numbers. And we see that is the case. So 370 are plus plus plus, and 350 are foo, bar, baz. Now, in the example problems, you'll see the chapter problems, we don't have them arranged like this, and you need to find the allele arrangement. So let's say if these were the highest, if this was 370 and this was 350, then that would suggest that the arrangement in the parent is actually like this, plus plus baz and foo, bar, plus. But that's not the case. Our original prediction of plus 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 on one chromosome, foo, bar, baz, must be accurate because these progeny with wild type for each gene or mutant for each gene are found in the highest numbers. This arrangement must be accurate. Okay, so now that we know which arrangement is accurate, I'm going to jump right to the shortcut method for determining which allele is in the middle. Okay, we confirmed that this is accurate right here. So what's the shortcut? Well, find the progeny with phenotypes that are represented, find the phenotypes that occur in the lowest numbers. Let me rephrase that. The phenotype combination that occurs in the lowest numbers. So that's plus bar Baz 9, foo plus 11. Now remember, in our previous video, we said double crossovers must occur in the lowest frequency, or always occur. Double crossover gametes are always the least frequent type of gametes formed by an individual. They're always least frequent than either single crossover class alone and the non-crossover class. So these guys right here must have resulted from a double crossover. Okay, so we know this is the correct arrangement of alleles. We do not know if bar is in the middle. If bar is in the middle, then, and we are correct, this is the right order, well, what we need to do is predict the double crossover gametes, the genotypes of the double crossover gametes formed by this individual, assuming bar is in the middle. And how do we do that? In a previous video, we said we just flip bar, or flip the gene in the middle. So bar will move up, plus will move down. So if this is true, this is the arrangement of alleles, which we determined to be true. If bar is also in the middle, then one of the double crossover gametes should be that genotype, and the other one should be that genotype. Okay, so these gametes should form progeny with these phenotypes, and they should be found in the lowest number if we are correct, if bar is in the middle. Let's see, plus bar plus, where is that? Plus bar plus, we see 38 of those. And foo plus Baz, we see 42 of those. So it looks like our prediction is wrong. Bar cannot be in the middle because it doesn't correctly predict the double crossover gametes in the progeny that have resulted from a double crossover. So we need to predict a different gene for the middle. So let's try this one. The arrangement of the alleles does not change, and I'm going to put foo in the middle. And I'm going to put bar over here, and I'm going to put Baz over here. Now, these guys right here, they're found in the lowest numbers, so let's call these the DCO offspring. They're the offspring that resulted from a double crossover in the heterozygous parent. Now, we know the arrangement of alleles is correct, right? Because these are the progeny, the offspring that resulted from non-crossovers. The pluses together, bar, foo, and Baz together, down here. Now, if this is the correct order, with foo in the middle, then the double crossover progeny, or gametes, should result when foo moves up and plus moves down. So we should have one double crossover genotype, should look like this, plus foo plus, and the other one should be bar plus Baz. So these should be found in the lowest number if our prediction is accurate. Now let's see. Bar plus and Baz are together. Bar plus and Baz are together. Okay, good. So this one is found in 9 of the offspring. Plus foo plus. Plus foo plus. It's found in 11 of the offspring. Okay, so we figured out what gene is in the middle. Foo is in the middle. Because when we have foo in the middle, we are able to accurately predict the double crossover gametes, which lead to the production of these phenotype combinations that are found in the lowest numbers. Okay, so foo is in the middle. Great. So that's how the 3-point cross works, and that's how you can tell what gene is in the middle. And we can do more now that we know foo is in the middle. And there are other ways you can go around to figure out what gene is in the middle. I think that's the shortcut method. So what is the shortcut method? find correct allele arrangement in the heterozygous parent, and then, let me see, determine which order produces, or correctly predicts, I should say, correctly predicts DCO offspring. And so what are the DCO offspring? Well, you look at the offspring table, see those that are formed least frequently, those are the DCO offspring. And foo in the middle is the only one that will correctly predict these to be the DCO offspring, progeny with these phenotypes to be the DCO offspring. Okay, once we know that, what can we do? Well, it helps to map it out. We can determine the distances between the genes flanking foo, which we know now is in the middle. So bar, foo, baz. Now what we can do is predict the correct classes for single crossover 1, crossover 2, and the double crossover. And that's simple. So here's a non-crossover here. One of the genotypes in the non-crossover class, bar, foo, and baz. The second genotype in the non-crossover class. Then I'm going to cross over here. I like to keep this anchored, this plus, I'll keep this anchored here. So foo and baz will move up. And plus will move down. So this is a single crossover class. We'll do the other single crossover class. Keep this anchored here and just flip baz up, plus down. So that's a single crossover class. And then the double crossover. What do we like to do to figure that out? Just flip foo up. And then that plus in the middle down. Okay, double crossovers. Okay, now we need to put the progeny numbers by each one of these gametes. Because these progeny numbers, they're essentially telling us that in parent 1, 92 of this plus baz gamete was formed. Plus baz, where's that? Right here. So this one's 92. And 88 of the foo bar plus was formed. Okay, now let's see. Plus foo plus, where's that? Plus foo plus, 11. Bar plus baz, 9. Plus, 370. Foo bar baz, 350. Plus foo baz, where is that? Plus foo baz. You guys probably see it already. Oh, here we go. 42 and plus bar plus 30. Ooh, hold on a second. Oh, I messed up here, didn't I? Bar should be there. Bar plus 38. Okay, now that we have these numbers, we can figure out the distances between bar, foo, and baz. Now that this is our map, our genetic map, and it's accurate for foo being in the middle. And now we can get the distances between bar and foo and baz. So the distance is going to be the total number of progeny. This should add up to 1,000. So the total number of progeny that resulted from a single crossover in this interval. Well, all the progeny that resulted from gametes that resulted from crossovers in this interval. So I shouldn't say crossover, single crossover, because we actually have to add single crossovers and double crossovers, right? Because to get a double crossover, you need a crossover here and over here. So to calculate this distance, we need to count crossovers in one of the single crossover intervals plus the double crossovers. So what genotypes result from a crossover between bar and foo? So I'm going to go back up here, and if there's a crossover here, that means foo and baz move up, plus and plus move down. So I need to find where we have plus and foo and baz together. Here we go, right here. Plus foo baz. So I'm going to call this single crossover 1, and we're going to call this interval 1, and I'm going to call this single crossover 1. So to get this distance, I need to add single crossover 1, which is going to be 80 individuals, plus the double crossovers, which is 20 individuals, which is 100 of the progeny resulted from a crossover between bar and foo in the heterozygous parent. 100 divided by 1,000 equals 0.1, and we can convert that to a percent by multiplying it by 100, and we have 10% of the progeny, or 10 centimorgans. So this distance is 10 centimorgans. Okay, what about this distance? Well, this distance right here is going to be just the other crossover, which I'm going to call single crossover 2, again, plus the double crossovers, right? So adding these up will give us all of the offspring that resulted from a crossover between foo and baz in the heterozygous parent. So let's just check that this is SCO2. How do we know this is SCO2? Well, I decided that this one is what we're going to call 2. And if we anchored, these are pluses. It's getting pretty messy up here. These are pluses. So a crossover here causes baz to go up and plus to go down. So we need to look for the genotype that has 2 pluses and baz next to each other. 2 pluses and baz. Okay, good. And then this is the reciprocal. So 92 plus 88 is 180. 180 single crossovers plus the double crossovers gives us 200. So 200 divided by 1,000 total equals 0.2. Multiply that by 100 gives us 20%. Okay, so the genetic distance between foo and baz is 20 centimorgans. And okay, well, yeah, that's it. That's it for the 3-point cross. And not only did we find that foo was in the middle, but using this progeny or offspring, sorry, I use progeny and offspring synonymously. I consider them to be synonyms. But using this offspring table, offspring phenotype table, we're able not only to determine what gene is in the middle, but the distance between the gene that's in the middle and the flanking genes, the genetic distances between the gene in the middle and the flanking genes. Okay, so we do have 2 example problems on the 3-point cross. I think they are, I don't have the numbers here, but there are 2 that were assigned, and there are solutions to those problems that I have typed up for you and provided on ReggieNet. So please try the problems on your own first, then look at the solutions. And I believe I even have some YouTube videos from a couple years ago where I walked students through the problems. And I may have taken a slightly different approach to solving those, but any approach works. And if all else fails, make sure to email me and set up a time to come to office hours or extra office hours. I think today is February 6th, 2020, and we have office hours here in about 45 minutes, so hopefully someone will stop by. Okay, well, I hope you understand that. Sorry again that the videos are a bit rushed, so again, to save time, I don't have time to edit them, don't have time for multiple takes. Hopefully moving forward in the future, I'll see which videos are the best and which videos need to be done. But for this semester, I hope what I'm doing now is at least helping some of you. Okay, well, if I don't see you in office hours, see you in class on Tuesday.